expect you to know, but I'm going to talk about some of the major animal groups. All right, so let's start with the clicker question. So get out your clickers. One of the characteristics of animals that they have is that they are heterotrophs. What does that mean? Click in if you have not clicked yet. I guess Fridays are harder to get up for less people here than usual. All right. One of the major ways that you can classify groups of organisms is through these metabolic terms. So we've been looking at a lot of autotrophs and heterotrophs and of various varieties as we've been going through this diversity of life. Heterotrophs, what a heterotroph is, it means that they have to get their organic compounds from another organism or from another source. Hetero means other, troph is eater, so they're other eaters, basically. So the correct answer here is B. They get their organic molecules from other organisms. So basically what that means is that there are no photosynthetic animals. There are no animals that can take carbon dioxide out of the air and turn it into organic compounds. They're going to be reliant on autotrophs for that. What type of heterotroph are animals? Chemoheterotroph, exactly. So animals are chemoheterotrophs. The chemo, it describes where they get their energy from. So the auto versus hetero is where do you get your carbon molecules from. The chemo versus photo is where do you get your energy from. So a chemoheterotroph, in order to get organic molecules from other compounds, and then they get their energy from breaking down those compounds. They get their energy from chemical reactions. Okay, that makes sense? Any questions about that? All right, just a little bit of a review on these terms. All right, so talk to your neighbor. What traits can you think of that makes an animal an animal? How do you know something is an animal?
Multicellular. No cell wall. Have internal digestion. Most can move.
Got the PowerPoint. All right, what kinds of things did you guys come up with? Not all animals have sexual reproduction. They are all multicellular. That's true. But like a lot of animals have asexual reproduction, and a lot of other things that aren't animals have sexual reproduction. So reproduction is not a terribly good way to determine whether that something is an animal. What else?
They go through gastrulation.
They do go through gastrulation. I'm not even sure I have that on the slide, but so we're going to talk about embryological development, and that's a pretty major step in embryological development for animals. Gastrulation is a stage in the embryonic development where a core forms that eventually is going to become the digestive system. What else? Did you have, you were just, no, you weren't raising your hands. What else?
Internal digestion.
Yep, they have internal digestion, in contrast to fungi, which are also heterotrophs,
External.
but they have external digestion. So animals, nearly all animals have internal digestion. There's a couple of weird ones, but nearly all of them have internal digestion. What else?
Homeostasis.
Yeah, pretty much all living
All
things have
living
some sort of
things.
homeostasis. Again, it's not going to help you tell animals from non-animals. What do you know about the cells of animals?
cell
versus,
wall.
like, the cells of plants?
No cell wall. Right
Yeah,
here.
no cell wall. Yeah, so animal cells do not have a cell wall. I'm trying to remember if there's anything else on here. I don't actually remember. Oh, we got it. Oh, we missed the moving. Yeah, this is one that most animals actually move, which is in contrast to a lot of other organisms, although there are some protists that can move. Again, protist is kind of a, like your euglena. Remember you saw your euglena? They were the little swimmers in your algae. they were moving. But animals are kind of unique in that most of them are pretty mobile. All right, so those are some of the traits of these animals. And so the first thing we're gonna do is, but there's a lot of diversity. Like, there's a whole bunch of things that are considered animals, everything from things that are relatively sedentary, like the sea squirt, through, things that you're much more familiar with, like a mammal, like a tiger, insects, there's a bunch of microscopic animals. We're gonna be looking at a microscopic animal next week in lab. We're going to be feeding our yeast to rotifers, which are a microscopic animal. They're really fun. So there's a lot of diversity of animals. So we're going to be looking at the classification of animals and how we group them into some major groups. And there's two types of things that are really, really important for placing animals into different groups. And those are developmental patterns, so how do they develop from an embryo through to an adult form, and also some stuff about the body plan, so how's the layout of the body. So we're going to walk through some of these different patterns so that we can compare some different organisms. So we're going to start with developmental patterns. All right, so one of these developmental patterns is the number of tissue layers early in development. So we're talking about embryonic tissue layers. There's basically two options. We have diploblastic animals. Diploblastic animals have two tissue layers, two cell layers. They have an ectoderm and they have an endoderm. Derm just means like tissue or skin. So if you have ectoderm, what do you think that is? Ecto. What does that sound like?
Outside?
Outside. So you have the outside tissue, and you have the endoderm. What's that sound like?
Inside.
Inside. You have the inside tissue. Ectoderm and endoderm. Okay. So an example of a diploblastic group are the cnidarians, or the group that contains the sea anemones, jellyfish, corals. Those guys are all diploblastic. Okay. Cnidarian starts with a C. The C is silent. Yeah, it's CNI, but it's pronounced cnidarian. So that's one form. The other option would be a triploblastic form. What do you think that's going to be? Three cell layers. Okay, and if you have outside, inside, what do you think the next cell layer is going to be? Middle. That's exactly right. So it's very predictable.
Meso.
You have triploblastic organisms. They have an ectoderm, an endoderm, and a mesoderm. So the mesoderm is between the ectoderm and the endoderm. Most other animals, well, at least all of the other groups of animals that you need to know are triploblastic. So there's actually a lot more triploblastic animals than there are diploblastic animals. One thing to note though is that sponges, which are an animal, are weird. And I'm going to be coming back to this a lot. Sponges are a very, very strange animal. Sponges have no true distinct cell layers. They just have different cell types. And actually you can take a sponge and put it in a blender and blend it up into individual cells and then pour it back into seawater, and they will find each other and form back into a sponge. So yeah, they're really cool, but kind of weird. They're super weird, actually. So we'll talk more about sponges in a little bit. All right, so that's one pattern that you can look at. Is the animal diploblastic or is it triploblastic? And this is, again, just remember this is referring to embryonic tissue layers. The next pattern is a pattern of gastrulation. This pattern applies only to triploblastic animals, so animals that have 3 embryonic tissue layers. There's some patterns in what happens during gastrulation. So gastrulation, early in embryonic development, you have a sphere of cells, that's called a blastula. That blastula then has the formation of the start of the gut, all right? So remember, if you're thinking about a gut, we have a mouth, we have an anus, and we're basically a tube, right? So in order for that tube to form, you have to have an infolding that starts. This is called the blastopore. And this process of the infolding is called gastrulation. So the blastopore folds in, it goes all the way across through the middle of the embryo, and eventually that infolding is what's going to turn into the gut. Okay, that makes sense? But you always start with this first infolding called the blastopore. Now, the distinction is on what that blastopore turns into. So one option is that the blastopore, the opening that forms from the blastopore, becomes the mouth of the organism. Because remember, this is forming the digestive system. We have two openings, we have a mouth and an anus. So if that infolding becomes the mouth, then we're looking at a protostome. Breaking down the word here, proto means first, and stoma is mouth. So these ones are forming mouth first. Okay, that makes sense? So that infold, and then eventually this will come all the way across the embryo and form another opening, and that second opening would be the anus. All right. So some examples of animals that are protostomes are the mollusks, mollusca. The mollusks include things like snails, octopi, clams, all of those types of things are all mollusks. And the arthropods. Arthropods are anything with a crunchy exoskeleton, so insects, lobsters, shrimps, isopods, if you know what an isopod is. Those are all like arthropods. Okay? Questions about this so far? All right. The other option is that the blastopore becomes the anus instead of becoming the mouth. So that first infolding becomes the anus, and then the second opening will become the mouth. Those are called deuterostomes. So stome again means mouth, deutero means second. So the mouth forms second. If you want a funny mnemonic, you could call them deuterostomes because the anus forms first. But the actual meaning is the mouth forms second. I just find that amusing because I am also kind of have an inner 12-year-old, so don't mind me. So things that are deuterostomes include the chordates. Chordates are the group that includes vertebrates, so we are a vertebrate, but also some other organisms as well that don't actually have vertebra. So the chordates and also the echinoderms. Echinoderms include sea stars, sea urchins, and sea cucumbers, if you guys are familiar with those critters. Okay? Any questions about this bit? All right, so that's the next pattern. Okay, one final pattern of development has to do with what we call cleavage. It's not that kind of cleavage. Cleavage refers to the first few cell divisions of the fertilized egg. So when an egg is initially fertilized, it's a single cell. And then you have the first cleavage. That cell divides from one cell into two cells. The second cleavage, it divides from 2 into 4 cells. And then the third cleavage, it divides from 4 into 8 cells. So in particular, we are interested in that third cleavage, where you're going from 4 cells into 8 cells. So one way that this can happen is what's called spiral cleavage. So you have this 4-cell embryo. When each of these 4 cells divides in half, instead of stacking right on top of each other, they're slightly rotated, kind of like they're in a spiral. So you can see that the lines between the cells are not lined up on top of each other. They're a little bit rotated, okay? So that would be spiral cleavage, right? And some organisms that have spiral cleavage, again, the mollusks have spiral cleavage, also the annelids. Annelids are the segmented worms. So earthworms, leeches are annelid worms, okay? Questions about that? All right. The other option for cleavage is that you have radial cleavage. And so in radial cleavage, those new cells are stacked directly on top of the old cells so that the lines between, or the divisions between the cells are right on top of each other here, like this. So directly on top instead of off to the side a little bit. So that's radial cleavage. Chordates and echinoderms have radial cleavage. Okay? Okay. All right, so those are our embryos. embryonic traits, which is good to go back to that for a second, is that they tend to be conserved. What that means, conserved, is that they don't evolve very quickly and they don't change very often. So you can compare embryonic traits between things that are very distantly related and may look really, really different as adults, but the way that they look as embryos can actually tell you a lot about how related they are, okay? So embryonic traits can be really, really useful for lumping organisms together into taxonomic groups. All right, moving now on into the general body plan or the structure of the animal. So we're going to talk about a few different features that you might use to describe what an animal looks like. The first one is symmetry. So symmetry is basically most animals have, most but not all animals have symmetry. What that means is that there is a plane through which you can divide the animal that will divide that animal into two halves that are mirror images of each other. That's what it means to be symmetrical, okay? And there's two major types of symmetry. There's radial symmetry. So in radial symmetry, you can think of radial symmetry like a pizza or a pie. There are a lot of different ways you can divide that in half and create two equal halves, all right? So that's radial symmetry. You can divide it in a bunch of ways and you get two equal halves. Bilateral symmetry is another really common type of symmetry. In bilateral symmetry, there is only one plane in which you can divide the organism where you get two halves that are mirror images of each other. So if you look at a human, for example, if you divide us in this plane, you get two mirror images. But if you divide us in this plane, you don't. Or if you divide us in this plane, you don't. So that's bilateral symmetry, all right? And bilateral symmetry is really, really common among animals. So why has that evolved? What's so great? Think about why it might be useful to have bilateral symmetry, to not have radial symmetry. Then you can talk to your neighbor.
Is there a slide?
No, but I can, oh, yeah. Movement,
What does it say?
says movement and civilization.
Yeah, I would think it would have to do something with the way they move.
Yeah. Like get to tight spaces.
Another way to think about this is, what does this guy have that this guy doesn't have?
Eyes?
Among other things.
A brain. A brain? Does brain have something to do with it?
All right, so what kinds of things are you coming up with? What are your ideas for what's so great about bilateral symmetry?
I'd say it just makes it, you can make that organism more complicated and structured.
There is a level of complexity that does tend to come with it. So what is one thing that you notice that's different between these guys?
One has an eye.
It was what?
One has an eye.
It does not. Okay, so the eye up here, this is a sensory organ, right? It's acquiring information about the environment. What do you have to do with that information? Process it. So you usually have your sensory organs in close proximity to a sensory processing unit, namely your brain. Having the sensory organs and the central processing unit in one location is called cephalization. It's basically the formation of a head region. That's what the head is. The head is an area that's centralized sensory information and centralized processing of that sensory information. What else can that fish do that the jellyfish can't do? Well, swim. This guy can swim. He kind of drifts around. He doesn't have directionality, right? He can move, but there's not really a forward or a backwards when you're a jellyfish, right? You can have much more efficient movement if you're bilateral, because you can have specializations for the forward end and different specializations for the backward end, because you now have directionality, right? Whereas this guy doesn't have a forward end and a backward end, right? So what you tend to see is a combination of these two things. You do have more efficient movement, and you have this specialization. If you're moving through your environment, you're moving through your environment and you have a directionality to your movement, where do you want your sensory organs? In the front. As you're moving through your environment, you want your sensory organs, I'm a fish, you want your sensory organs to encounter the environment first, right? Because it doesn't make sense to swim into something and then have your sensory organs in the back end, right? That's not helpful. So you tend to see that the concentration of sensory organs and the concentration of processing of that sensory information is in the front end of the animal. All right? That makes sense? Okay. So that's what the advantage of bilateral symmetry is. More efficient movement and specialization of different parts of the body, particularly the front end of the body for sensory reception and data processing, which is cephalization. Okay. Questions about that? All right. Are there any asymmetrical animals? Yes. Like what? Sponges are weird.
Anemone?
Sponges are the only truly asymmetrical animal. And as I said before, sponges are extremely strange. They do have specialized cell types, but they don't have true tissues. And they can form all kinds of weird shapes that you cannot divide in a single plane and create two halves. Now you may be thinking, what about a coral? Because a coral might have a shape like this too. But a coral is not one animal. A coral is hundreds and hundreds of tiny little individual animals, each with their own little shell. And if you look at those individual little animals within the coral, they have radial symmetry, so they are symmetrical. They actually look like little mini tiny sea anemones. They just happen to live in colonies that are asymmetrical. All right, so sponges are weird, not surprisingly. All right, that brings us to the next categorization here, which has to do with body cavities. This is something that sometimes people find a little bit confusing. There's this concept of internal body cavities that exist and we're not talking about the cavity within your digestive system. So the digestive system, there is a cavity there, a gap. But we're talking about spaces in the body that are filled with fluid that are between the digestive system and the outer wall, okay? So if you don't have one of these fluid-filled body cavities, you are what we call an acoelomoid. A means without. So they don't have a coelom. And again, I don't know why it's pronounced with an S, even though it looks spelled like it should be pronounced colon, but it's pronounced coelom. Don't ask me how these things get, I don't know. English is a very strange language, and biology English is even weirder. But yeah, so we have these acoelomates. Acoelomates, they have the ectoderm, the mesoderm, the endoderm, no fluid-filled body cavities there. An example of an acoelomate are the flatworms. If you guys have ever heard of a planaria, they're a very cute little flatworm. They look like that. They look like they look like they're happy little cute worms, but I don't know that they have enough brains to actually be happy. But they're strangely cute for a worm. You'll get to play with planaria when you take zoology. Dr. Fran always has a planaria experiment in zoology, so that's fun thing. So that's an example of an acoelomate. A pseudocoelomate has a fluid-filled body cavity. That fluid-filled body cavity exists between the mesoderm. So we're talking about ectoderm, mesoderm, endoderm. So these terms, again, only apply to triploblastic animals. You can't apply these terms to diploblastic animals. It only works for triploblastic animals. And the fluid-filled body cavity exists between the mesoderm and the endoderm, okay? So the mesoderm is usually what makes up the musculature of the animal that allows the animal to move. And so have you guys ever had one of those long skinny balloons and you squeeze it in one part and it moves the liquid to the other parts of the balloon? Like if you have like a long skinny balloon filled with water, you squeeze it in one part and it makes another part extend. So that fluid can actually help an animal to move by contracting muscles around the fluid. In a pseudocoelomate, because they only have musculature outside that fluid-filled cavity, they don't have very precise movement. So an example of a pseudocoelomate are the roundworms or the nematodes. I showed you guys the fungus that eats the nematodes. And nematodes really can only do one kind of movement. They can do this. And they wiggle through space, right? And they can't really do anything more precise than that. They just kind of, they contract their muscles and they go wee, 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 back and forth, all right? If you have a true coelom, so this is a coelomate, a true coelom by definition is a fluid-filled body cavity that is surrounded on all sides by mesoderm. That means you have musculature on the inside of the fluid-filled cavity and on the outside of the fluid-filled cavity, okay? This allows you much more precise movement. The annelid worms have true coeloms. If you've ever watched an earthworm try to move, you might watch them and they will extend the piece of their body out, put it on the ground, and then pull themselves along, right? That extending out, what they're doing is they're contracting the muscles around that fluid-filled body cavity. That causes it to kind of, it's like they call it a hydrostatic skeleton. That causes that liquid to move, which allows them to stretch out towards the front. And then they can pull their musculature to contract themselves up again. All right? So these guys actually have much more precise movement than a nematode has because they have that true coelom instead of a pseudocoelom. Pseudo means fake. So these ones have a fluid-filled body cavity, but it's not a true coelom because it's not completely surrounded by mesoderm. Yes? It's called a nematode. Yeah, we'll be going through the different, after we talk about these different traits, we'll be going through the different critters, and I'll show you some pictures of the different organisms that are in these different groups. But nematoda is the name of the group, but you could also call them roundworms or nematodes. Other questions?
Which one are we?
We are a true coelomate. We also have a fluid-filled cavity that's completely surrounded by by mesoderm tissue. The cavity that supports all of our organs, our abdominal cavity, that's our coelom.
Okay.
Now we don't rely on our fluid-filled cavity for movement because we have an internal skeleton with muscles attached to it. And so that's how vertebrates move. So we move in a different way, but we do have a true coelom. Other questions? Okay. All right, and then the last thing is segmentation. So a lot of animals have repeating segments, so different parts of the body that are specialized for particular functions. And this allows for a lot of adaptation. So a lot of the animals that have been most successful actually have this segmentation pattern. Because if you're segmented, you can have different parts of your body that are specialized for different things. So you might have a segment that's specialized for movement. You might have a segment that's specialized for sensory perception. You might have a segment that's specialized for, in this case, defense. There's a lot of these organisms that have segmentation. So the ones that are really clear are things like the arthropods. The arthropods have distinct segments. If you look at an insect, they have the head, the thorax, the abdomen, they're segmented. The annelid worms are also sometimes called the segmented worms. This is an aquatic annelid worm, but earthworms are also an annelid worm. And they have specialization of each individual segment. Technically, vertebrates also have some segmentation. If you look at our vertebrae, that is an example of segmentation. We have a repeating structure that moves from well, we don't have tails, but if we had tails, it would go from our head all the way back to the tail. And those different, the different vertebrae are specialized for different things. So your cervical vertebrae are specialized for different functions than your lumbar vertebrae are. So that's another thing that you can look at. All right, any other questions about any of this stuff so far? All right, so we're now going to, based on the, I forgot I had the clicker in there, based on the previous information, and particularly think about the embryonic information, what group of animals do you think that chordates, which is the group that includes the vertebrates and ourselves, what group of other animals do you think we are most closely related to?
I have no idea. What do you think? I'm going. That's what I went with. You're a coelomate.
Yeah.
All right,
Either
click
that
in
or
if you haven't
B.
clicked.
Did you switch your answer?
All right, so if you think back to what we were just talking about, particularly the embryonic things, which I said, remember, they change very slowly, so they can tell you what's related. The thing that we are actually most related to is echinoderms. So, which is really surprising. Things like starfish and sea urchins don't look like they'd be terribly closely related to us, but echinoderms are deuterostomes, vertebrates are deuterostomes. Echinoderms have radial cleavage. The chordates also have radial cleavage. They were both true coelomates. Another thing that we have in common is that we both have calcium-based endoskeletons. Have you guys ever seen a sand dollar? They've got that calcium-based endoskeleton, or even a sea star or a sun star. This is a sun star. They also have a calcium-based endoskeleton, just like we do. So there's a bunch of similarities, even though they look very, very, very different from chordates. That's the thing that we are most closely related to. So here's a phylogenetic tree of animals. So animals are a monophyletic group. The common ancestor of all animals is considered an animal, and all descendants of that common ancestor are also animals. Okay, so that's a monophyletic group. The sponges, as I said, sponges are weird. They're the most distantly related of the animals. They diverged first. And so they have no true tissues. Then you have the evolution of specialized tissue types. And you have your radially symmetrical things. The reason that these are next to each other is because we're not 100% sure of the relationship here. That's what this means, that you're not 100% sure of the relationship. These are your radially symmetrical diploblastic animals over here. These are your bilaterally symmetrical triploblastic animals. So lots of triploblastic animals. One little group of acoelomates that come out over here. And then you have your body cavities. You can be a protostome or a deuterostome. So here we are, the deuterostomes. The only thing in the deuterostome are the chordates and the echinoderms. So that's one of the major reasons how you know that we're related. And then within the protostomes, we're going to look at two different groups, the ecdysozoans and the lophotrochozoans, which, if you have never said the word lophotrochozoan, it's actually a really fun word to say. You just have to practice it, lophotrochozoan. So I'm not going to ask you guys to know all of these animal groups. I would like you to know that arthropods, and actually this isn't even all of the animal groups that exist, but I would like you to know the arthropods, the nematodes, the platyhelminthes, or the flatworms, the annelids, the mollusks, chordates, echinoderms, cnidarians, and sponges. So we'll go through each of those groups individually. So if I don't have a slide in the PowerPoint of the group, you don't have to worry about memorizing that particular group, because there's a bunch of other groups, and I don't want you to memorize everything. You'll learn everything when you take zoology, but we're going to hit the major groups. And these are the groups that have the most organisms in them. That's why I'm focusing on these groups. They're the most common and the most abundant of the animal groups. Okay. We are gonna play with some rotifers in lab, but that's actually not a group that I'm going to require you guys to know. But just there they are. So when you see the rotifers, you can be like, ah, yes, they're a Lophotrochozoan. So you can just think that to yourself while you're playing with rotifers and laugh, maybe. All right, so let's go ahead and look at some of these. Oh, wait, before we look at those, let's talk about what animals evolved from. So animals evolved from a heterotrophic eukaryote that was similar to, we have these existing heterotrophic eukaryotes that are not considered animals, that are the closest living relative of animals, called choanoflagellates. And so these choanoflagellates are actually the closest living things. Remember that animals and fungi are closely related. So if you drew a branch for the tree, you'd have fungi over here. And then over here you'd have animals. And you'd have choanoflagellates. Right? So those are the closest living relatives. So that means what? If they're our closest living relatives, what does that mean? All right, click in if you haven't clicked. All right, so yeah, so if they're our closest living relative, then that means that animals and choanoflagellates share a common ancestor. That would be this thing right here. They've evolved from that common ancestor, so have animals. But the interesting thing about these choanoflagellates is that they can exist as individual cells, so they can exist as a single cellular organism. But some of them have simple multicellularity, where they live in these groups that are attached to a single cell. If you look at these cells, at the choanoflagellate cells, they actually look remarkably similar. Well, okay, this is where I was going. They look remarkably similar to, this is a sponge. See this cell here? Looks an awful lot like that, doesn't it? So they think that one of these colony lid-forming choanoflagellates is what eventually, or an ancestor of these colony-forming choanoflagellates is what eventually gave rise to the sponges. So remember, sponges are weird. They have these different individual cell types. Some of these cell types are called choanocytes, but they have other cell types that are different throughout their bodies, and these cells are organized in a way that allows the animal to function as a whole. So sponges are an animal that doesn't move, although the individual choanocytes do have flagella, and those flagella spin and draw water in through what are called the pores of the outside of the sponge. This is where the scientific name for the sponges comes from. The scientific name for sponges is porifera. So they've got all these pores along the sides of the sponge. The choanocytes are spinning, they're moving water, and so that draws water in through the pores. And then there are these cells that are kind of like amoeba-like cells that can engulf bits of food that are drawn in on the current of the water. So these guys are basically filter feeders. So the water gets drawn in through the pores on the side, goes out through the top of the sponge, and they're able to get their nutrition from these little particles that are in the water. All right. So here's our phylogenetic tree again. Remember, there's the choanoflagellates. They're not considered an animal. Sponges, however, are considered an animal. So this is the common ancestor of all animals. All animals are multicellular. All animals do have gastrulation. So they do start with that ball, and they start with that blastophore. They also all produce a protein called collagen. Collagen is the protein that is in your skin that makes your skin stretchy. It also makes your tendons and ligaments stretchy. So that for us, that's what collagen is super important for. But collagen is a protein that basically holds the bodies of animals together. Very, very important compound. So that's those are the traits that all animals have. You got your sponges over here, they're super weird, they don't have true tissues. And then you have the evolution of tissues, and you've got your cnidarians over here, which have radial symmetry, and your bilaterians over here, which have bilateral symmetry, and more complex organs and whatnot. Okay? Any questions about this so far? All right. So let's go ahead and go through some of these different groups. So the peripera, that's that group that's really different from all of the other animals. This is not a great picture, but there's a lot of diversity in sponges. There are sponges that incorporate silica into their structures. Those are called the glass sponges. And then there are a lot of, and actually, the reason that the sponge that you use to wash your dishes is called a sponge is because we used to actually harvest sponge animals and use them as sponges. In fact, you can still get like a fancy bath sponge that actually comes from a sponge. There are species of sponge that, like if you've got like a super fancy bath sponge, it actually probably comes from an animal. It's like the skeleton of that animal that you're using to wash yourself, which is kind of honestly a little bit weird. But, yeah, so this picture here, this is a glass sponge. It's not a great photo. But the glass sponges, of course, you would not use to wash yourself because they've got silica skeletons that are super scratchy and hard, and that would not be good. All right, so that's the peripera. Any questions about the peripera? Okay. Moving on to the cnidarians. So remember, the cnidarians are bilateral. Not bilateral. They have radial symmetry. They're diploblastic. They have two embryonic tissue layers. Another thing about cnidaria is that they do not have a digestive tract. They have what's called a gastrovascular cavity. They have only one opening to their digestive system. It acts as both the mouth and the anus. Food comes in and waste goes out from the same opening. All right. So basically, these guys are just big, empty sacs. Jellyfish are a really common cnidarian that you may have heard of, and sea anemones. Who's watched Finding Nemo? You know that Nemo lives in an anemone, right? Sea anemone. So these guys are more or less the same thing. It's just if you took a jellyfish and took the front half and stuck it to a rock, then you get a sea anemone. In fact, most jellyfish, or actually most sea anemones, go through what's called a medusa phase, where they're free floating and basically like a tiny jellyfish as a larvae, and then eventually when they mature, they stick themselves to a rock. And they go into what's called the polyp phase. So this is the polyp phase. They're a polyp as an adult, but they're a medusa as a juvenile. Cnidarians also have a special cell type called cnidae. The cnidae are basically like little mini cellular harpoons. And so they've got a structure that they can actually eject and stick into a potential prey item, and then they can retract and grab it and hold onto it. Has anyone been to an aquarium where you could touch sea anemones? You know how they stick to your finger? That's the little cnidae that they're actually they're like harpooning your finger and holding onto it. And if you were like a little shrimp or something, then they would bring that arm in, and this is their mouth right here in the middle. So that if they harpooned like a little shrimp or something, they could bring that in and put it into their mouth with their arm, which is kind of neat. So corals, this looks like an asymmetrical structure, but the individual coral animals, you can actually kind of see them here. That's one little animal right there. All right, and they look just like this, but much, much smaller. So they're basically like a teeny, teeny sea anemone, but they build these large skeletons around themselves, and then those skeletons form the corals. Any questions about cnidarians? Okay. All right, so that brings us into the bilateral animals. These are the bilateral triploblastic animals. And the next major division is between the deuterostomes. So remember, in the deuterostomes, the blastopore forms the anus, and in the protostomes, which are these guys down here, the blastopore becomes the mouth. And then within the protostomes, we're going to break them down further into ecdysozoans and lophotrochozoans. Again, I encourage you to practice the word lophotrochozoan. You'll have fun with it once you start saying it. All right, so we're going to start. We're actually going to go from bottom to top through these different categories. So we'll start with the lophotrochozoans. There are a bunch of lophotrochozoans. The only ones I need you to know are the annelids, which are the segmented worms, the mollusks, which are your snails, octopus, clams, and the platyhelminthes, or the flatworms. So those are the ones I'm going to ask you guys to know. These are the most common, the most abundant. The rest of these are somewhat less common. So let's start with the annelid worms. So the annelid worm that you're most familiar with is the earthworm. There are giant earthworms. They do exist, that can get up to 6 feet long. One of the places they live is in the Palouse Prairie in eastern Washington. They thought they'd gone extinct because Palouse Prairie is very heavily agricultural land, and if you plow, if you can imagine you're plowing the land and there's a 6-foot earthworm, you're going to cut that earthworm up into little bitty pieces. So they're only present in a few little patches of unplowed prairie that are over there, but they're still really cool. But there are some other things. There are tube worms, if you've ever heard of a tube worm. This is a tube worm outside of its tube, but tube worms are also annelid worms. These are a bunch of different types of marine worm. These are called, this one's called a polychaete worm, that are pretty cool as well. And then the other group that you might have heard of that are annelid worms are the leeches. And leeches are primarily aquatic, parasitic worms that feed on blood of vertebrates for the most part. A lot of them are specialized to feed on frogs and salamanders, but there are also terrestrial leeches. Have you guys ever heard of these? Land leeches. Where I did my PhD research in Australia, we had land leeches, and you'd be out in the forest, and they crawl along the ground, and the land leeches primarily target mammals and birds, and because of that, they're heat-seeking, because mammals and birds are warm-blooded. And so if you're in these places where there's a lot of land leeches and you look down, they're on the forest floor and they're coming for you. It's really creepy. But as blood-sucking parasites go, I have to say, these are my favorite blood-sucking parasite, because they don't carry any diseases, it does not hurt when they bite you, and they don't buzz around your face. So as blood-sucking parasites go, I'll take these guys over a lot of other blood-sucking parasites. The annelid worms in general are true coelomates. They're triploblastic. They're protostomes. They have segmentation. So all of those characteristics that we were talking about, those are some of the characteristics that the annelid worms have. Any questions about annelids? All right. Moving on to mollusks. The mollusks include all of the bivalves, so your clams, your mussels, your oysters, all of the snails and slugs, and also the squids and octopi. They're a really interesting, really diverse group. So, they've got some very, very simple organisms in the mollusks, but also some of these, if you're going to break them down to major groups, you have your bivalves. Bivalve means 2 shells, right? You have your gastropod. Gastropod means stomach foot. That's your snails and slugs. And then you have your cephalopod. Cephalopod means head foot. So, and the cephalopods are the most intelligent invertebrates. Octopi can learn by watching other octopi. They're extremely smart, they're delicious, and I have serious guilt about eating them, so I've actually stopped eating octopus because I feel bad because they're so smart. But there's a bunch of things that aren't octopus, like cuttlefish. Cuttlefish are another type of cephalopod. They have very complex communication, which they achieve by changing the color of their skin and by the position in which they hold their tentacles. Yeah. Bivalve. I once encountered a cuttlefish while snorkeling, and I was really, really excited. And I knew that they communicated by holding their tentacles and by changing their color. And so I decided I was going to try and communicate with this cuttlefish. So I came up to it and and it turned white and it backed away. So I don't know what I said, but it definitely didn't like it. So yeah, they're super cool. Again, true coelomates, bilateral symmetry. What else can we say about them? No segmentation, these guys. They do have spiral cleavage. That's about all I can say about the mollusks right now. Any questions about mollusks? And we'll end with this if anyone